Hello everyone, myself Anita from First MSc Microbiology, Sacred Heart College, Tirupattur. Today we are going to discuss about biochemical test. The main aim of biochemical test is used to measure the levels of bacterial enzymes which is interrupted accurately to identify the species of a bacteria producing that enzymes or not. Biochemical test is mainly classified into four types: enzyme-based biochemical test, carbohydrate-based test, and protein-based test and biochemical test IMViC. In enzyme-based test, it is classified into five types: catalase test, oxidase test, urease test, phenylalanine deaminase test, and coagulase test. In carbohydrate-based test, it is classified into three types: carbohydrate fermentation test, triple sugar ion, which is also known as starch hydrolysis. In protein based test, it is classified into two types. One is gelatin hydrolysis and another one is casein hydrolysis. Finally, the IMViC test, that is Indole test, methyl red test, Voges test, and citrate utilization test. Today, we are going to discuss deeply about IMViC test. First one is Indole test, and the medium used for Indole test is tryptone broth, and the indicator is Kovacs reagent or Eldridge reagent. Eldridge reagent is mainly used to differentiate anaerobic bacteria, and the principle of Indole test is in the culture medium the amino acid tryptophan is present, and this tryptophan is acted upon the enzyme tryptophanase, which is produced by the bacteria and converted into indole, pyruvic acid and ammonia. After 24 hours of incubation, by adding the Kovacs reagent or Eldridge reagent, it produces a red ring on the top. This is a simple schematic representation of indole test. The amino acid tryptophan present in the culture medium is acted upon the enzyme tryptophanase, which is produced by the bacteria and converted into indole, pyruvate and ammonia. The indole positive bacteria are E. coli, Proteus vulgaris and Vibrio cholerae, and indole negative bacteria is Klebsiella pneumoniae, Proteus mirabilis, Salmonella species and Shigella sonnei. Observation and results. After 24 hours incubation by adding Eldridge reagent or Kovacs reagent, the positive indole shows formation of pink to cherry red ring and the negative indole test shows no cherry red ring. This is indole positive which shows cherry red ring and indole negative that shows yellow ring. The next one is methyl red test. The medium used for methyl red test is EMA and VP broth. This EMA and VP broth is alkaline in nature which has higher than 7 pH and the indicator is methyl red. The main principle of methyl red test is whether the particular bacteria produces mixed acid fermentation when it is supplied to glucose. The mixed acid fermentation such as lactate, acetate, succinate, formate, ethanol as well as equal amounts of water and carbon dioxide. As I have already told that EMA and VP broth is alkaline in nature. When methyl red is added, it changes into acidic pH and shows red color. The methyl red test positive bacteria is E. coli and the methyl red test negative bacteria examples such as Klebsiella pneumoniae, Serratia marcescens and Vibrio cholerae. And the results. The positive EMA test shows red color ring and the negative EMA shows yellow color ring which has higher pH due to the fermentation of glucose. The next one is Voges test, that is VP test. The medium used for Voges test is EMA and VP broth as same as methyl red test. And the indicator is alpha naphthol. The main principle of VP test is that whether the particular bacteria produces acetoin when the glucose is fermented. Here, two reagents are used. One is VP reagent A and VP reagent B. In VP reagent A, it consists of 5% of alpha naphthol and VP reagent B consists of 40% of potassium hydroxide. These two reagents are added after incubation and produces pink color. The example of VP test positive bacteria is Klebsiella pneumoniae, Serratia marcescens and Vibrio cholerae, and VP negative bacteria is E. coli. Remember one thing that the MR positive is negative in VP and the MR negative is positive in VP test. The result in the positive VP test shows formation of pink color at the surface. This is a positive test, and negative VP test shows lack of pink color, and this is where VP negative test. And the final one is citrate utilization test. The medium used for citrate utilization test is cement citrate agar, which is green in color. And this cement citrate agar consists of two sources. One is carbon source and nitrogen source. The carbon source consists of citrate and the nitrogen source consists of ammonia. And the indicator is bromothiamyl blue. As I have already told that citrate is a carbon source and ammonium salt is a nitrogen source. This is a chemical formula for ammonium salts, that is NH4, H2 and PO4. The bacteria grow in this enzyme produces citrate permease, which is capable of converting citrate to pyruvate. The bacteria produces citrate permease enzyme and it is converted into citrate and pyruvate, which increases the alkalinity about 7.6. When the pH is turned into less, the bromothiamyl blue indicator, it shows green to blue color. This is a simple representation of citrate utilization test. The citrate which is present in the culture medium is acted upon the enzyme citrate permease, which is produced by the bacteria, is converted into ammonium salts and then to ammonia. The citrate positive bacteria is Klebsiella pneumoniae, Serratia marcescens and Vibrio vinificus, and citrate negative bacteria is E. coli, Salmonella species and Xylella species. And the result, positive citrate utilization shows color change from slant to green to blue. Green, this is a negative citrate utilization test which is changes into blue color, and this is positive citrate utilization test. And the negative citrate utilization shows no color changes, that remain same in green color. Thank you for listening.